It is my great pleasure to welcome Professor Richter-Levin. So unfortunately not at UCL, but within the UCL community. So Gal, I'm going to give a little bit of an introduction, but I think, if you want to add on anything, please do not hesitate. So Professor Gal Richter did his PhD in Israel at the Weizmann Institute. And I think it's straight after your PhD that you came to London, if I'm correct. And that's when you worked as a postdoctoral researcher with Tim Bliss at Mill Hill at the National Institute for Medical Research, which is now known as the Crick Institute. And that's where you met Steve Hunt, if I'm correct. And after that, you went back to Israel. And that's where you started leading the different behavioral neurosciences groups. So you're currently professor of behavioral neuroscience at the Sapol Department of Neurobiology and the School of Psychology at the University of Haifa. And you've also been, awarded and numerous prizes, and you've been the president of numerous prestigious societies within Israel, including the Israeli Society for Biological Psychiatry and the Israeli Society for Neuroscience. And today you're going to tell us about your recent work in the context of post-traumatic stress disorders. In particular, you're really interested into the individual responses to the experience of a traumatic event, and you're going to tell us how we can dissect out the individual, response using animal models, and how like a more complete behavioral analysis and a full phenotypic of the animal can lead us to have a better understanding of the neurobiology of PTSD. So without further ado, please, I give the floor to you. Thank you very much for accepting the invitation.
Thank you very much for inviting me. It's quite exciting to give have the opportunity to present this talk, and I'm looking forward to the discussions later. So yes, I'm going to talk about trying to understand individual variability in response to stress and trauma. And the fact that there is individual variability is actually well known. The only thing is that normally we try to ignore it or to mask it, and not to refer to it, or to refer to it as noise that we need to get rid of, where I want to try and show that it is actually very important, particularly if you want to kind of correlate behaviors and brain mechanisms. It is very important to pay attention to the individual differences, because this would make the story much more accurate. And it is easiest to present it or to demonstrate it using an extreme condition like exposure to trauma, or model of post-traumatic stress disorder. And since this is anyway what I'm studying, I would use this example to demonstrate that. So post-traumatic stress disorder is associated with the exposure to trauma. And the first criterion for diagnosing anyone for suffering from PTSD is that they have been exposed to a traumatic event. So much so that the thinking is that the trauma actually induces the pathology. And this line of thinking also controls the thinking about the animal models. So the issue in animal models is to expose animals to trauma and then see what the trauma was doing. So the basic structure of a study in animal models is to compare a group that is exposed to a traumatic experience, the average of that group, to the average of a group that has not been exposed. However, there is a problem with this model, and I want to demonstrate it in the following way. This is a graph from a paper about comparing two types of trauma, and it seems to be that there are some interesting differences between the intentional and non-intentional effects of trauma. And this is what the paper is about. However, what I saw in this graph, or in similar graph, is the part that is not shown. The fact that actually most individuals exposed to the trauma do not develop pathology. And if most individuals that are exposed to trauma do not develop the pathology, it means that the trauma itself is not a sufficient condition to induce the pathology. It means that there must be some predisposing factors that actually enable the trauma to lead to pathology. And so if we want to understand the mechanisms of the pathology, we need to understand the role of these additional factors. But also, it means that working with averages is problematic, because if you take the average of the group that was exposed here, the contribution of those who actually suffer from the pathology is very small, and makes it very difficult for you to identify what is related to the pathology. So we try to work on both ways. One way is to try and work with risk factors and introduce risk factors to the model and try to understand what the risk factors may contribute to the pathology. And the other, which is not mutually exclusive, as you will see, was to try and develop ways of differentiating between those that are exposed and are affected and those that are exposed and are not affected. And I want to describe to you work on both directions. I will start with the risk factors and move to this dissociation between affected and unaffected animals. So there could be several risk factors. We focused mainly on one, on the exposure during childhood to adversities, which are considered to be a risk factor for developing psychopathologies such as PTSD and depression later in life. And when you come to work with the rodents, the most accepted model at the time for early life stress was some form of maternal deprivation. But we felt that maternal deprivation at the first 2 weeks of the life of the pups is too early. It's not childhood. It is a much earlier developmental stage. So we moved to juvenile or post-weaning pre-puberty stage, which is much greater similarity to what we think about when we think about childhood in terms of behavior, in terms of brain development, etc. And then the idea is quite simple: to expose the animals to some form of adversities in their childhood, and then to come back to these animals in their young adulthood life and expose them to some trauma. And the expectation is, of course, that the effect of the adulthood trauma would be much more severe if it comes on the background of exposure to the juvenile stress. And this is just one example. This is work that has been published quite a while ago. So what you see here is, first of all, that if we compare the effect, in this case of the startle response, between a group that was exposed to trauma only in adulthood to a group that was exposed to the same trauma in adulthood but of the background of juvenile stress, then you can see that they respond more than the ones without the background. And even more interesting is the fact that in the group that was exposed to trauma only in adulthood, 3 weeks later, there was a recovery, and there was no such recovery in the group that was exposed to the same adulthood trauma, but on the background of the juvenile stress. And work like this led us to the model we are using, which is the juvenile plus adulthood stress model, where we expose animals to the juvenile procedure. You can see here the types of procedures we use, but I can tell you there's already several labs that have adopted this model, and it doesn't need to follow exactly these procedures. Any type of stressor that, and these are not very severe stressors, but the important thing is are the dates of exposure. And then we wait 1 month or 1.5 months to young adulthood, expose animals to trauma in adulthood, and then test them 1 week, 2 weeks, or even 1 month after the trauma, and look for the effects. And here is an example of a study where we looked at the animals 1 month after the trauma, the adulthood trauma, and you can see that the adulthood trauma by itself had some effects on anxiety, on activity, but the same measures were much more severe if the same trauma was on the background of the exposure to the juvenile stress before. This made us very happy, and we felt that we have a good model at hand. By the way, it follows findings in humans, where in groups of, for example, Vietnam soldiers, the reports on childhood adversities in those developed PTSD is much higher than the reports in the group of soldiers that were not developing PTSD. And now we have the model, and we could start looking at mechanisms that are involved. And we have some specific interest in GABA and in interneurons and the way they modulate principal cells. This is based on findings from many laboratories, including ourselves, showing that indeed exposure to stress modulates different aspects of GABA functioning. And one tool that we have is when you record in the hippocampal dentate gyrus, you can use relatively simple electrophysiology, and you have several protocols, which I will not go into the details, but these protocols actually emphasize different aspects of feed-forward, feedback, inhibition, and the interaction between interneurons and the granule cells in the region, and it's relatively convenient to measure. And we found that if you induce the juvenile stress plus adulthood trauma, the result is that there are changes in this local circuit activity, but very specific. So not all protocols are affected, only specific protocols, suggesting that only specific subpopulations of interneurons are responsive to this manipulation. This fits very well the general concept that there are different types, different subpopulations of interneurons, and they may carry different roles and may be affected in different ways by experiences. But we wanted to be more specific and to go in greater detail to which interneurons are involved. And if you work with mice, it is you have more tools because you have genetic lines that you could kind of target specific types of interneurons. Working with rats, as we do, for reasons that I would be happy to discuss, but we are working with rats. You don't have those genetic tools, so what we do is we use viral vectors, and we want to target specific GABA-related targets in the region of interest. We are lucky to have a very fruitful collaboration with Hans-Jürgen Volkmar and Martin Krebel from Tübingen, Germany. With them, we found that there are specific proteins that are associated with the clustering of GABAergic synapses at specific subregions of the granule cells. So, for example, neurofacin is responsible to interacting with Jefferin for enabling clustering of GABA receptors, specifically at the axon initial segment, which means that it affects the functioning of the subpopulation of interneurons, the chandelier cells, which affect the axon initial segment. And it does not affect other interneurons. And we decided to see the impact of reducing the expression of neurofacin, so using a viral vector into the dentate gyrus in juvenility, so instead of actually exposing the animals to juvenile stress, we found the same pattern of electrical changes, so that the protocol that was affected, the only protocol that was affected as a result of this manipulation, was the same protocol that we found to be affected following juvenile and adulthood stress. But what is more interesting to me is the fact that this kind of relatively mild modulation of dentate gyrus activity by changing the modulation of the neurophasin affected interneurons was sufficient to induce a phenotype, was sufficient to cause an impairment in the learning under stress task, the shuttle box activity avoidance task, in a way that was similar to what we found following exposure of animals to juvenile stress and testing them in adulthood. This suggests that relatively moderate changes or alterations in the activity within the dentate gyrus are sufficient to introduce kind of individual variability in the response of the animals to challenges such as this task. And we have a reason for being interested in the dentate gyrus, because the dentate gyrus demonstrates a type of, I would say, of a riddle. On the one hand, it is the less interesting part of the hippocampus. It is not the area where you have the place cells, etc. And so it is considered to be like the gateway of information into the hippocampus. But on the other hand, this is the region where you have neurogenesis. And we know that neurogenesis is not so much found in other areas except for the olfactory bulb. And that neurogenesis is affected by exposure to stress. And that if you use treatments that remove or reduce the chronic stress symptoms, you also see recovery of neurogenesis. And neurogenesis happens not in the hippocampus, but in the dentate gyrus of the hippocampus. And we don't actually know what is the role of the neurogenesis in that respect, and why is it so correlated with this kind of responsiveness to stress. And we, coming from a completely different angle, found something that was also focused on the dentate gyrus. We have been working on the effect of stress and of amygdala priming on plasticity in the hippocampus. So the idea was that there were many reports talking about stress suppressing LTP, plasticity of the type of LTP in the hippocampus. But it turns out that all those studies were actually focused on looking at LTP and CA1. We started to look at both CA1 and dentate gyrus in vivo in animals, exposing them to different protocols of priming of the amygdala and different protocols of stress. And we found that indeed CA1 was sensitive to stress and to amygdala priming, but in a monotonous way. It didn't really matter what protocol of stress or what priming paradigm we used to activate the amygdala. It suppressed a potentiation in the CA1. Whereas in the dentate gyrus was not only sensitive to stress or to the priming, but to what exactly the priming of the stress is, because you could get different outcomes. And this means that when the stressful level of the experience increases, the contribution of the dentate gyrus to what would be the outcome of processing in the hippocampus increases. It means that, in fact, under these conditions, the dentate gyrus has a much more pivotal role in defining the outcome. And this means, again, that small changes in the dentate gyrus could mean significant differences in the ability to cope with learning under stress. But I want now to move to this other direction of working with the affected and unaffected animals, because As we just mentioned, PTSD is associated with the exposure to trauma, and the classical way of modeling it is by comparing the average exposed group to the average non-exposed group. And this way ignores this individual variability that exists in both humans and animals, as we mentioned before. And this led us to understand that we need to develop a way to separate between those animals that were exposed to the trauma, but were behaviorally affected by it, from those who were exposed to the same trauma, but for some reasons were not affected behaviorally, were not affected by it. And this work was done by a group of past students in the lab, and at some stage in collaboration with Hagit Cohen and Yossi Zohar. And we came up with what we call the behavioral profiling, and I will try to briefly explain what we try to do. So basically what we do is we expose the animals to whatever procedure, traumatic procedure that we choose to use. And then 1 week, 2 weeks, 1 month afterwards, we expose the animals to a battery of tests. And it is important that it be a battery of tests and looking at various behavioral parameters, because we found out that as convenient as it may be to use only one parameter to characterize the animals, this is not accurate enough. An animal could, just by chance, have a bad day or a good day on one behavioral parameter. But a healthy animal would not accumulate many negative symptoms, and a pathological animal may show a behavioral parameter that would be okay, but would not accumulate many good kind of behavioral parameters. So basically what we do is we look at the average response of the control group, the group that was not exposed to anything, because the control group represents normal behavior. This has the advantage of not defining whether this is depressive, anxiety, or whatever symptom. This is just not normal behavior. And we define the average, and we define deviations from the average. So for example, in this study, we used one standard deviation away from the average is not a normal behavior. And then we come back to each animal, including the control animals, and to each behavioral parameter, and we ask whether this parameter is within the normal range or out of the normal range. And then we count the number of out of the normal range parameters. And an animal has to, as I said, accumulate several abnormal behaviors in order to be considered as affected. And this leads you to be able to separate between the animals that are really affected and the animals that are resilient. And this enables you actually to much better separate between mechanisms that are associated with the pathology and mechanisms that are associated with being resilient. And interestingly, we see that the proportions of affected animals are very similar in these animal models to what we know in humans. So if you only have a trauma in adulthood, you have about 25% of the animals that become affected. If you add a risk factor, like the juvenile stress that we discussed, you can see a dramatic increase in the proportion of affected animals. But you can still see that even in this situation, you have a significant proportion of the population that has been exposed to the juvenile and to the adulthood stress, and their behavior is still fine, emphasizing the need to separate between these groups. But as I told my students, this separation is based on behavioral symptoms. And if we really want to trust the functionality of this dissociation, behavioral dissociation, we need to find differences in the brain that would be correlated with these definitions of being affected or not affected. And this is what we did next, for example, here, where we looked at the activation, map activation of cells in different brain regions. We used CIFOS to identify activated cells, and we used double labeling with GAD67 to differentiate between excitatory activation and inhibitory activation. And you don't need much statistics to see that the map of activation of the animals that were exposed but not affected, the upper layer, is much different from that of the animals that were exposed to the same trauma, but were significantly affected by it, indicating that indeed the separation into the affected and unaffected is critical to understanding what happens in the brain, because if you work with the group averages of the group, you mix these two types of brains. And I want to give you an example of a study where we compared the conclusions you may get working with the group average to the conclusions you may get when working with this behavioral profiling. So we are interested in GABA and interneurons, as mentioned, and some years ago, we found that exposure to stress results in changes in the level of expression of some GABA-related proteins, including the GABA receptor alpha subunits. So we decided to come back to that, now using the behavioral profiling approach. Basically, we had three groups: a control group, a group that was exposed to stress only in adulthood, where we expect a relatively low proportion of affected animals, and a group that is exposed to the same trauma in adulthood, but on the background of juvenile stress. This is, of course, the group that exhibits the most significant symptoms compared to the others. And we found in that group quite a significant elevation of expression of the alpha-1 subunit of the GABA receptor in both different parts of the hippocampus and in the BLA of the amygdala. And because this comes from the group that shows the highest levels of symptoms, it is relatively easy to come to the conclusion that this elevation of alpha-1 probably is related to the pathology. And that if we want to help these animals, we need to find a way to reduce the expression of alpha-1 or to prevent the elevation in its expression. However, now moving to analyzing the same set of data using the behavioral protocols, the behavioral profiling protocol, and separating between affected and unaffected, we found out that the elevation in alpha-1 came mainly from animals that were exposed to the trauma but could cope and actually were defined as unaffected, suggesting exactly the opposite, suggesting that the elevation in alpha-1 is probably part of the mechanism of establishing resilience, of coping with the situation. And that if we want to help those animals, we need to find a way to enhance the expression of alpha-1. This is how critical differentiating between the affected and the unaffected animals could be. And I want to give another example from a more recent work, which we did in collaboration with Jelena Rodolevich from the Albert Einstein College of Medicine. In this case, we were interested in changes in microRNAs, and the fact that microRNAs are responsive to stress is not new, and there are many publications indicating changes in different types, in different microRNAs. But we referred specifically to a study by the group of Jelena, where they showed that exposure of animals in adulthood to a stressor resulted in changes in the expression of several microRNAs, which are modulators of several GABA-related proteins. So we decided to come back to that But now look at the changes of those mirrors that they found to be responsive, but in different hippocampal subregions, to look at the behavioral profiling, so to differentiate between changes in affected and unaffected animals, and also to look at the difference between males and females. So basically, the animals were exposed to the juvenile class adulthood stress, and then behavioral profiling took place where we characterized the animals as affected or not affected, and then we looked at the expression of the microRNAs. We found affected animals in both males and females, and we found several changes in those, the expression of those microRNAs in different brain regions that we looked at. We focus here mainly on the dorsal and ventral hippocampus, and to make it easier, I kind of focus on the main changes that we found. The main changes that we found in males were elevation in the expression of miR33 and miR144 in different locations. The 33 was in the ventral dentate gyrus, the miR144 in the dorsal dentate gyrus of resilient individuals, suggesting that this elevation in the miRs is somehow part of the mechanisms of resilience. In females, we found similar elevation in the same, with the same miRs. However, in different location, both miR33 and 144 were elevated significantly, but in ventral CA1 and in ventral CA1 of affected animals, suggesting that this elevation is actually part of pathological mechanism, a marked difference between the males and females, suggesting that mechanisms of resilience and mechanisms of vulnerability may be very different between males and females. To kind of enhance that finding, because it was so robust, we repeated this study in a different way. What we now did was to try and block the activity of those microRNAs in the locations where we found the increase. And the idea is that if you have an increase that is related to resilience in males, then blocking the activity should impair resilience, should increase the number or the proportion of affected animals. And the opposite in females. If we block the elevation related to pathology, not the elevation, but the activity of the elevated miRs in these areas, we should actually enhance resilience. And this is what we did here. So we indeed found that if we block the activity of the miRs, the miR144 in the dorsal dentate gyrus and the 33 in the ventral dentate gyrus of males, we found impaired resilience, meaning an increase in the proportion of affected animals. And the opposite in females, this blocking of miR33 and 144 in the ventral CA1 resulted in increase in the proportion of animals that were characterized as resilient. Final example that I want to give relates again to an attempt to differentiate between which interneurons are involved. And this time we tried to look at different interneurons by actually looking at the co-localized neuropeptides. It is somewhat strange that many people work on neuropeptides, many people work on interneurons, but in fact, most of the neuropeptides are actually co-expressed in GABAergic interneurons. And different subpopulations of interneurons are or can be defined by the neuropeptides that they actually co-express. So we started to look at that. This is in collaboration with Oliver Stock from Magdeburg and Anna Albert as postdoc in my lab. She's now in Magdeburg as well. She has her own lab there. And Stavrig was the PhD student. And we looked at changes in neuropeptide expressions in the different parts of the hippocampus. Here we compared fear conditioning as a kind of a mild stressor in adulthood to fear conditioning on the background of juvenile stress. And we indeed found that there were more affected animals in the fear conditioning on the background of juvenile stress, as we discussed earlier in the talk. If we look at the average results, we don't see much, but if we separate the animals to affected and unaffected, we found that there was a significant increase in the activation of neuropeptide Y containing interneurons, specifically in the dorsal dentate gyrus. We repeated this experiment now using a different trauma in adulthood, but basically the juvenile stress plus adulthood trauma as before, again, separating between affected and unaffected animals and looking at different neuropeptides. And I will go directly to neuropeptide Y. Again, we find a specific increase in the expression of neuropeptide Y, specifically in the dorsal dentate gyrus of the unaffected or resilient animals. And again, as we discussed with the mirrors, if this is indeed related to resilience, then reducing the expression of neuropeptide Y can be expected specifically in the dorsal dentate gyrus, could be expected to impair stress resilience, meaning that we should see increase in the proportion of affected animals. And so this is what we did. We used the viral vector into the dorsal dentate gyrus to reduce the expression of neuropeptide Y, and the result was that there was a significant increase in the proportion of affected animals in this group that the neuropeptide Y was affected or was impaired in expression, indicating that it is sufficient to impair the expression of neuropeptide Y, specifically in the dorsal dentate gyrus again, to have a phenotype, to have impaired resilience. Trying to summarize what I've presented to you into what we can learn about individual variability and mechanisms of stress resilience. First of all, this individual variability is not noise. We need to learn to understand and to work with the individual variability, because individual variability differentiates between those animals that can cope and those animals that actually would develop pathology. It is very important that we would differentiate between these two. And we need to be able to separate between animals that have been exposed to the trauma and are affected by it, and animals that have been exposed to the trauma and somehow manage to cope with it and behaviorally not be affected. And I mentioned behaviorally not be affected because it's not true that they're not affected. You see changes, they are not like control animals that were not exposed. There's a kind of active resilience, if you want, but they are affected in a way that enables them to continue coping in terms of the behavioral performance. And we need to find ways to work with the differentiating between these animals. The behavioral profiling, as we do, is not perfect, but it is one way that brings us closer to what we need to do, and it's a convenient way, and it's an easy way to adapt in different laboratories because it is a concept. It does not require specific behavioral tests It doesn't require specific trauma. It does not require a specific animal. It just requires using this kind of approach. And I would be happy to discuss with anyone who is interested how you do it in different contexts. Another thing that I think is of importance is I talked about microRNAs, neuropeptides, GABA receptor, subunit, etc. There are many changes at many levels in many different brain areas. The real description of what happens is very complex. It's very much multi-level, multi-site, multi-genes, etc. But despite that, there seems to be a possibility to identify points of intervention, which means that it is sufficient to intervene, and even relatively mildly intervene in these points, in these targets, in these regions, to get a phenotype, to change the level of resilience or the level of vulnerability. Probably because you induce a change in the network. And this is, I guess, what is our challenge for the future. It is to identify such points of intervention and ways to intervene specifically in those points in order to change the fate, to change the outcome of the exposure to trauma. And one such spot, which turned out to be very significant in this respect of individual variability to resilience, is surprisingly the dentate gyrus. It is, we have accumulating findings to suggest that the dentate gyrus has a kind of a unique role, particularly when stress and trauma come into place, to kind of direct the outcome of these experience. It is not that the dentate gyrus is the location of resilience or vulnerability, but it is such an intervention spot that is of critical importance. And this is it. I want to thank different collaborators, funding agencies, my current group, and thank you for listening.
Thank you so much. Gives us a lot to think about. So I will start with a couple of questions and then I'll open the floor for questions. So my first question will be about like looking for, in a way, an outcome measure to really give us an indication of what has been the impact of the trauma exposure to the individual animal, right? And you talked about the behavior and about the molecular biology, but I was surprised that you never talked about potentially looking at stress hormone levels. Do you think that would tell, have you looked at it? And would that tell us anything? Is the hormone response would be an indication of the extent of the trauma exposure or not? Or it depends when you're looking. Can it help us in any way?
I have to be politically correct in that respect, but I think that there's no such thing as stress hormones. There are hormones that participate in the response to stress. They are not specifically stress hormones. They are more metabolic hormones. They are to some level correlated with what happens, but are not accurate. You know, if you think about, just think about the timing of things, okay? So if you look, you know, I like very much to look at the Nature Geographic Wild, and you see these encounters between animals. It takes maybe a minute. In that minute, cortisol, corticosterone has not yet come up. So the whole event occurs without having cortisol or corticosterone elevated in the blood. You have the parasympathetic response, you have the CRH release, and many other neurotransmitters working in the brain. You have a response, and it is not what we call the stress hormones.
Okay, but there's still an elevation of cortisol following the exposure to stress, right?
Well, cortisol has actually, if you think about it, cortisol, which comes out after 10 or 15 minutes, it has a very important role. if the risk is still there, if the problem was not solved, then it helps transforming the metabolic picture into one that enables longer lasting struggling. Okay, preserving energy there, releasing the possibility of energy there, trying to start the metabolism of fats and peptides to be a source of energy, etc. It changes the scene. If the risk is no longer there, then actually corticosterone suppresses the stress response. Right? This is what it does. It suppresses the release of CRH, it suppresses the release of GnRH, it has its negative feedback. So it has the role of actually ending the response if there is no need for it. Or elongating it, but in a different way, in a way that would be more suitable for a long response.
Coping behavior.
Yes. So it is relevant. It is part of, if you measure it, you measure a proxy of what happens. But it is, I would not call it stress response, because then you would expect something different.
Okay, sure. But in a way, so that has never helped you separate your animals into the resilient versus susceptible.
What you could do, and some people have done this, is to actually look not at the elevation, but at the relaxation. The rate of relaxation could be more informative of adrenaline, of corticosterone. The rate of relaxation may tell you more about the ability of the animal to come back to.
Sure. The issue we're having with all our animal model is how many times do you want to take blood from an animal that's already been subjected to stress, right? So
yes.
Okay, thank you. I'll keep my other questions because I know I'll be able to talk to you later. So Claire, please, if you have a question.
Thanks, Sandrine. I totally agree with you. Go about cortisol. I mean, all Celia's work shows that clearly cortisol is essential for coping with stress, rather than causing the harmful effects of it, which is why people need cortisol supplements post-operatively, for instance, because it improves therapeutic outcome. But I just want to pick up on your point about, I mean, very interesting work, by the way, absolutely fascinating.
Thank you.
Amazing findings. But to go back to something I mentioned earlier, I noticed, I mean, you're focusing on individual vulnerabilities, but what you measure experimentally is changes in group means, or differences in group means. And from that, you build a theory on whether any of these factors are beneficial or harmful. Now, from those predictions, if you then dig into the individual animals in the groups and look at correlations between the neurochemical measure of interest and the behavior, then you should be able to consolidate your hypothesis, because they would show either a positive correlation or a negative correlation, depending on what the group mean suggested. But there is a possibility that actually vulnerability and resolving, coping with the stress are actually on completely different neuronal pathways, mechanisms, and that what you see in the group differences, where you can reduce vulnerability or the stress response, that's actually recruiting a different mechanism from those which you see in individuals, which you test with just looking for whether it's a positive or negative correlation. Have you tried doing that?
So first of all, I want to agree with you that we eventually fall into working with averages, but I would say in a kind of a more accurate way, because we do, on the one hand, differentiate subgroups, but then we use these subgroups to look at averages. And if technically it was possible, it would have been much better, of course, to work with individuals. But this means that we would need to be able to look at thousands of parameters and their meaning. And we are not able to do that at the moment. This would mean that we would need to be able to tailor a treatment to each and every individual according to this complex combination of thousands of interactions between different mechanisms, which is something that we are not able to do at the moment. So yes, if you ask me, how does the system work? It works individually, and the elevation, for example, in NPY, which may protect some individuals, may not be helpful at all in some individuals. Okay, if you just kind of isolate this single one. And coming back to the average means that in many individuals, the elevation of NPY in the dentate gyrus is helpful. Not that in each individual it would be helpful, because the specific, the very individual context of that individual may not benefit from elevation of an NPY. I completely agree. But this is not something that we can work with. So what we try to do is to kind of relate to the fact that there are individual differences, but bring it to a level that would be workable. And this is basically what we find, that on the one hand, we and other laboratories talk about many targets and many changes in many brain areas. And I trust all those researchers that look at different things than what I do. And so I assume that in each animal, in each individual, there are so many changes. And I cannot manipulate them. I cannot describe a picture of what happens with all these changes. But I also find that these specific interventions, this kind of subgrouping and working with averages, but averages of a more homogeneous groups, and intervening in those homogeneous groups gives me a higher level of success of intervention. This means that I come closer to identifying something that is sufficiently general to help most of the individuals. I'm sure that even under these conditions, you would find some individuals that would not respond to such a manipulation, but there would be much fewer of those. So I completely agree with you on the one hand. On practicality, I think that we are not able really to work in this level. We do have some correlations. We do show some correlations which suggest that this indeed, as you described, this could be a more variable picture than what we try to kind of define. But in practicality, we do not work with that.
Can I just go back onto that point, because I was actually the question I was going to ask you. You know, I was thinking obviously all these should correlate. It's all not a linear, but a distribution of individuals. And for me, actually, when I think about resilience and vulnerability, I just think of them as two ends of the spectrum. So even though I completely agree with you that if you want to think about like clinical implication, it's easier for us to deal in the population thinking about groups. But do you think you could reanalyze your data maybe with 3 groups? Do you think there's such a dissection of like, could you be like a major amount of the population that we just like average in the middle, and then actually resilience and vulnerability being like pulling the other hands of the spectrum? Or we just exactly split into, not exactly split, but more like a resilient or more like a susceptible individual?
The only reason we can split it like this is because we look at the very narrow window of what we want the animals to do. Because resilience, you could add to it resilience to what? In our life, you know, someone can be very resilient to being a businessman, but not being able to work out relations. Someone could be, you know, it can be very resilient in one community of people with a culture of certain type, and not being able to manage in another society with different culture or something like this. It is much more complicated. We can only kind of I would say shape some rules that would be more effective than not referring to individual differences at all, but it is not really that we can go down in any practical way to the individual differences, because this is too rich for our technical abilities.
No, fair enough. Steve.
Yeah, thanks, Gal. That was really very interesting. I was just interested in this. I'm looking at Sandrine at the moment. I don't know. You clicked on. Anyway, I'm interested in this figure of resilience and vulnerability of about 25%, which is very similar to the vulnerability to cocaine self-administration. This addiction, this 25%, 17 to 25% seems a very consistent percentage. And given that I presume your rats are group housed, I mean, how does status map onto this vulnerability resilience picture?
How which status?
The alphas, the alpha males,
okay.
the slaves or whatever they are, worker bees.
This is extremely interesting question, the social context of things, which I should say that so far we have not worked with. So this is a kind of an additional factor that we have not yet dissected out. We have invested a lot of money now to set up a kind of a social cage where you can group animals that are identified individually, and you can start to look at the social behavior as well as at the later outcome. And unfortunately, we have been affected by the war in Ukraine, because they do not get the parts and they did not yet complete the it's a true story. But this, I agree that this could be important. This is why we are trying to set it up, so that instead of following the animals, most pharmaceutical companies use maybe the elevated plasmids and the outcome of one variable, which I think is really not informative at all. We are trying to use several tasks, several tests, several behavioral parameters, which I think is better, but it is still looking at the very, very brief snapshots of behavior of the animals and not having the social context.
Yeah.
So this is what we are trying to add now, a system where you could actually look at the behavior of the animals over several days, actually, collect the data, and then find a way to analyze it and try to see if we can have a more accurate description of the effects of, for example, the social hierarchy and other aspects of social interactions, and how this affects. And we could, if we have this cage, we can measure it before and after exposure to trauma. So we could see the differences of animals that have been characterized before.
Yeah.
But we are still waiting for this cage.
I mean, how do you work out status of an animal in the cage, in the group?
Again, the normal or the more accepted way of doing this is not the way we want to do, because the way that it is done is, again, you kind of gear the animals to a certain behavior. And what we want to do is to let the animals run. You have standard tests of hierarchy that you can use. We want to learn from analyzing the data. So you would have a huge set of data from the behavior of the animals, and you start to measure things like encounters, who is leading, who is pushing the one out, etc., etc. And you would need to kind of dissect from these behaviors, but you would have many such behaviors, who is the more dominant animal and who is the less dominant. And we could compare that to standard tests of dominance. But we want to kind of be able to pick it up from a more elaborated way of looking at the animals, particularly in order to have a more kind of natural way of defining the animal.
Thank you.
Well, Gal, thank you so much for giving us this fantastic talk.